I am leading the DNA repair group here at IFOM, and my lab is working on the 3 R's of life, which are DNA replication, DNA recombination, and DNA repair. Our genetic material is constantly exposed to endogenous and environmental damaging agents. And also, as we age, our DNA undergoes degeneration, a process that has been associated with genetic instability and tumorigenesis. In order to maintain genome stability, cells have evolved and are equipped with a tapestry of sophisticated and interconnected DNA repair mechanisms and DNA damage surveillance systems. So in the lab, we are really interested in understanding how cells are able to repair and tolerate DNA damage, how DNA recombination can rescue different replication problems, and in general, how these different DNA repair mechanisms are coupled with other cellular processes such as cell cycle transitions, chromosome replication, chromosome structure, and so forth. In order to tackle these issues, in the lab we are using different approaches that include genetic, genomic, cytogenetic, and biochemical techniques. But also I would like to say that a feature of our lab is to study the DNA structure of the intermediates that facilitate certain chromosome metabolism and DNA repair processes. We are also using different model systems, in principle cellular model systems. Also because what people do not appreciate perhaps enough is that cancer cells often behave like single organisms, so they are not so well integrated with the cells in the tissue, rather they care more about their own division and proliferation. So, as I said, we are using different model systems that include the single-cell organism, budding yeast, Saccharomyces cerevisiae, and also vertebrate and mammalian cell lines, such as chicken and mouse and human cell lines. Now, you might be surprised that budding yeast is used, I should say, successfully in a cancer research institute, but the truth is that DNA has been exposed to damage from the beginning of times, and because of this, DNA repair mechanisms appeared quite early on during evolution. So because of the very strong evolutionary conservation of the DNA repair pathways, what we learn even from our work in simple organisms such as budding yeast is applicable to understanding what's going on in human cells as well. As I was saying before, our work is focused on basic and fundamental questions related to chromosome biology, but these questions are also important for understanding cancer biology in general. So now it is becoming apparent that the genetic instability that drives tumorigenesis is often caused by errors in the DNA repair apparatus or by failures of the DNA damage surveillance system. We know that certain tumors respond well to certain categories of drugs, whereas others don't, and we do not always understand what is the DNA repair defect of certain cells. So, in principle, cancers record about 6 mutations, it is said, for genomic instability. But we know that even single mutations in DNA repair genes can make individuals cancer-prone. And it is really becoming clear that the DNA repair defect of cancer cells can be targeted. We have successful examples in the future for this. And by understanding the molecular mechanisms underlying the general or specific defects behind tumorigenesis is extremely important. I think that the work we are doing in the lab can really increase our knowledge and open new avenues for cancer therapy and human disease management.